what we have done so far is we have looked at the basics, right? And we have looked at glycolysis, gluconeogenesis, pentose phosphate pathway, and how glucose enter the cell. So now we will look about regulation. So how things are regulated. And so regulation is really important because a lot of things are happening within the cells, right? Many reactions are happening. And all of those processes have to be tightly regulated so that we like we maintain a homeostasis. So regulation is a sense of homeostasis where with the purpose is to make sure that the relative concentrations of metabolites remain more or less constant. Okay, so the reactions that are happening, you can compare them to the traffic in LA or New York, where there are so many cars, so many paths, so many vehicles are moving at the same time. And so really the purpose is to maintain that flow of traffic, right? So that's what is happening inside us, is we're trying to maintain the flow of traffic. And in the biochemical sense, that means we are maintaining the relative concentration of metabolites so that nothing is getting exceedingly high or nothing is getting exceedingly low. And if it does happen, then there are mechanisms which will restore the balance. So that is the principle of regulations. And we'll see that many of the enzymes, how they're regulated, and there are different mechanisms by which they're regulated. So that's the thing we will look at now. And so the main purpose is to maintain the flow of metabolites, to sustain homeostasis. They need to be adjusted depending on the metabolic need of the body. So for example, some reactions must be accelerated, some reactions must be decelerated depending on, again, what metabolic state of the body is. So there are different mechanisms by which this happens. One of them we have already looked at, which is feedback inhibition. We have looked at where did we look at feedback inhibition, right? So NADPH inhibits this G6P dehydrogenase by feedback inhibition, and the thought there is that if you have enough NADPH, then you're using it for anabolic processes. Then don't do pentose phosphate pathway. Rather, use store G6P, right? Store as glycogen. That's the whole point there. So we have seen feedback inhibition. The other type of regulation, transcription or translation level, meaning that promote the synthesis of an enzyme or inhibit the synthesis of an enzyme. So here we're looking at transcription or translation level, where you overexpress the cell or don't express the cell, overexpress the protein or the enzyme of interest, or don't express it to control the activity of that enzyme. Then there are other ways, for example, that there are different types of regulatory molecules. And so we will look at ATP, AMP, so how they actually regulate. So they will bind to different allostatic sites, of the enzyme, which will either activate or deactivate depending on the metabolic need of the body at that particular point. So the other way is by hormonal regulation, where we're secreting type of hormone to promote one type of pathway versus other. For example, insulin. So insulin typically will upregulate the activity of phosphatases, whereas glucagon will upregulate the activity of kinase. So the way it happens is by phosphorylation or dephosphorylation. And so protein phosphorylation happens at which amino acid site specifically? Alcohol. So examples will be what? Tyrosine, threonine, serine. So in general, we are going to see these types of mechanisms by which enzyme activity is regulated. And so when you are fed state, we have high glucose level, right? But when you are starved, then your glucose level is low. And so there are processes which will make sure that release glucose sent to brain. So for example, gluconeogenesis will be promoted under starved condition. And so because we have low glucose, so we've got to find a way to release glucose so that brain can use it for fuel for glycolysis and things like that. On the other hand, when you have high glucose, which is right after meal, then there will be enough ATP produced from this process, but also whatever excess, store it as glycogen, right? So that's the other mechanism by which this will happen. So there are two states, one is fed state, one is starved state, and depending on the type of, so usually insulin will be here, right? And starved states will be glucagon, because glucagon will promote processes which will increase blood glucose level, right? So make ways to find ways to make ATP for the body because we are running low on fuel. So it happens at points where reactions are far from equilibrium. So most of the reactions that happen within us are near equilibrium. Okay, so the forward and reverse processes can be easily adjusted by a change in the concentration of the substrate of the product. But regulation will happen mostly at the sites where the process will be irreversible. Okay, let's look at an example. So we have A, let's say A going to B and B going to C and C going to D. So these are the rates. Forward rate here is going to be, let's say 10.01 here, 0.01. B to C, let's do forward 200, 190, and then this one, let's do 500 to 450, 490. It means that all of these steps are happening at a net rate of 10, meaning that all of them are happening at a balanced way so that nothing is very high, nothing is very low, right? This is what the purpose is, to maintain this constant flow of metabolites, constant flux of metabolites through different pathways. So this rate really satisfies that all of them are being made at relatively same rate, right? A to B is 10, B to C is 10, C to D is 10. So everything is happy. There's all fine. So if you look at the ratio of these, so 200 to 190, and then 500 to 490, they're slightly greater than 1, right? Only slightly greater than 1. That means that change in concentration of any of these can easily readjust the rates, right? Because see that is near, all of them are near equilibrium, right? So the small change in concentration in either one of them can readjust that process to restore the balance. But if you look at the ratio here, in A to B, what's the ratio? 10 over, so how much is that? 1,000-fold. So now we see that the forward rate here going from A to B is 1,000-fold higher than the reverse rate. So clearly, if you change the concentrations of A and B for this pathway, it will not be easy to restore the balance, right? Because the rates are very, very different, right? The ratio, relative rates are very different than these guys. So therefore, this process must be highly regulated so that nothing goes out of the hand. So this will be tightly regulated. A to B will be the most tightly regulated. And so that's the principle of regulation. So this is clearly far from equilibrium. 1000 is not near equilibrium. This is very far from equilibrium. So this step must be tightly regulated. And in fact, that's what happens. Irreversible steps are mostly tightly regulated. What it also means that if the same substrates are regulated by different enzymes, then those steps will be most tightly regulated. We look at two things. One is high ATP and the other one is high AMP. So when do we have high ATP? Yeah, in the fed state we have high ATP, right? Then the ATP will be not consumed until I mean, it's constantly being consumed because we're working, right? So, but the rate of consumption is going to be slower than rate of production. But when you are starving, right, or we're doing exercise, right, then we are spending that ATP and we are making a lot of AMP. So it turns out the relative ratio of ATP and AMP, or rather AMP over ATP, is an indicator of which enzymes will be regulated versus not. So typically, high ATP will promote anabolic processes, right? Already we know that. And high AMP will promote processes that produces ATP. So in other words, release blood glucose, right? So 10% change in ATP typically will trigger about 500% change in AMP. So this is why AMP is an allosteric regulator of many enzymes. Okay, and high AMP will trigger glucagon pathways, which will make sure we're not doing energy consuming processes, but rather promote processes which will release blood glucose in the. So if you look at the relative concentrations of how ATP and AMP concentrations are maintained in the cell, ATP. So before an event, if it is 5 and after it's 4.5, but then similar process in AMP will be 0.1, but here it will be 0.6. So here this will be about 10%. This will be about 500%. Yeah. So ATP, if you have high ATP, then we are going to do anabolic processes, right? Because any synthetic work, so anything you synthesize or like muscle exercise or doing work basically is consuming ATP. And if you do anabolic processes, then your ATP is going to drop, your AMP is going to increase, right? Or when you're starving, under low nutrient conditions, you don't have high ATP, your AMP is high. So if you have high AMP, then what must happen is that we must release glucose into the blood, right? So that brain can take it and other tissues can take it. So liver will release glucose. It will promote glycogen breakdown. We'll look at later next semester. And many other processes which will increase glucose in the blood so that other tissues can take it. So high AMP means that somehow we need to release glucose, make more glucose, release it into the blood. We are starving or we did exercise, right? Either one of these conditions, either during exercise or starving conditions, AMP's levels are high. But how does AMP actually activate those processes? So that's what we're going to look at next. Okay. And so there is a kinase, AMPK means AMP activated kinase. And again, they respond to the relative ratio of AMP to ATP. This is a kinase, so that's an enzyme by itself, which is activated by AMP. Once this is activated, then that will activate a target enzyme by phosphorylating either the serine, threonine, or tyrosine, which can either make it active or inactive depending on the context. But phosphorylation is one key mechanism by which enzyme activity is regulated. So AMPK will be activated by binding of AMP to its regulatory sites. Once it's activated, it will take ATP and use the PI of ATP to phosphorylate the target proteins. Different tissues have different needs, and different tissues respond differently to the metabolic need of the body. So liver is the sort of the big daddy of the whole body. It will give glucose, whatever, to all the tissues whenever they need it. Muscle is just like it just wants to take glucose all the time because muscles are always working. Muscle contraction needs a lot of ATP. Hypothalamus of the brain, for example, that triggers the signal that we have to eat, right? So AMP has different functions, or it differentially controls the function of different tissues. So that's something we need to keep in mind. So arrows going this way, meaning these are the effectors which are activating AMPK. And then we draw a bunch of arrows going the other way, which will represent what is the effect of activating AMP kinase. So in the muscle, it will uptake or upregulate fatty acid oxidation, because when you oxidize fatty acid, we get a lot of ATP. Once we have glucose, we can do glycolysis to make ATP to replenish the lost ATP concentrations. In the brain, the signal is that we need to eat. In the heart, it will also do fatty acid oxidation and glycolysis. So when you have high AMP, the goal is to actually increase ATP somehow. And these processes is what is going to make more ATP. And it will inhibit all the energy consuming processes, right? For example, anything synthesis, if you synthesize anything, all of those synthetic processes or anabolic processes will be down-regulated by high AMP, right? Adipose tissue where fat is stored, so it will inhibit fatty acid synthesis. A similar effect also happens in the liver. And in fact, the other process that will inhibit is it will inhibit the pancreatic beta cells to secrete insulin, so that basically you're trying to make glucose available for the tissues. We have high AMP when we are not eaten, we are starving, or we have exercised. So we have depleted ATP. We need to find a way then to restore ATP, increase ATP concentration. And that can be done by promoting glycolysis or promoting fatty acid oxidation, right? So that we can get ATP. And of course, when you don't have ATP, then you don't do the processes which requires ATP. So anabolic processes, right? For example. So that's the big picture of how AMP activated kinases work.